and they will answer questions. And so I'm not going to waste time doing a lot of introduction, so we can catch up a bit. The first speaker is Adrian Thrasher from University College London, who's a pioneer in the treatment by genome editing of genetically inherited diseases, of which you've heard several times. There are thousands, and he's been heavily involved in this for a number of years. He's going to tell us some of his latest information. Thanks, Adrian.
Okay, thanks very much, Richard, and thank you to the organisers for asking me. Where's everybody gone? I tell you, 18 years ago, when we started treating patients by somatic gene therapy, not editing, then we used to get that sort of thing, but you know, that's how things change. So, my job is to just to walk you through some of our clinical experience of somatic gene therapy, because I think there are a lot of learnings from that process that are applicable to gene editing. And I'm focusing on the hematopoietic space because that's my area of interest. And I'll end up with some comments about gene editing and where we might see that being applicable in this space too. So our interest is, well, my clinical interest is in inherited immune deficiencies, which come in all sorts of shapes and sizes, but in the severest forms, these children will die within the first year of life from unusual infections or common infections, which disseminate because the children are born without any immune systems. Now, the history of treatment of these severest diseases is interesting because you go back to 1968 and the first successful non-twin transplant, bone marrow transplant, was performed in the X-linked form of severe combined immune deficiency. Before that, every child died, as I said, usually within the first year of life. And now, today, we not only have newborn screening for this disease, but we also have advanced bone marrow transplantation technologies. We have gene therapy, which is almost becoming a standard of care for some of these diseases, as I'll show you, so that we would expect to cure now the majority of children. So we've gone over that admittedly long time period from an invariably fatal disease to one that we would expect to cure. Now, the reason that the immune deficiencies have been favored targets for developing gene therapies and are currently targets for developing gene editing is because often the corrected cells have a selective advantage. Okay, so if you can efficiently gene modify or edit stem cell populations, then the selective survival and growth advantage means that they can fill out the immune system with functional cells. So on that basis, many years ago now, around about 2000, our group in London and Alain Fischer's group in Paris conducted a clinical trial using gamma retroviral vectors. This is old technology. So very simple gamma retroviral vectors in the X-linked form of severe combined immune deficiency, and modified bone marrow cells from these children after considerable informed consent, and infused the cells back into the patients without any preconditioning. Now, the alternative for these children was a highly mismatched transplant, and at that time, we would have expected 30 to 40% of those children to die going through the procedure. And what you'll see from this graph is that the majority of the children responded very well. So they start with no T cells, which is the most important cell in terms of fighting viral infections, for example. And they develop T cells pretty much into the normal range for age-matched controls, enabling them to fight off any infections. And we go now 18 years down the line, and we're still seeing these patients in clinic, and they still have a very robust immune system, it's highly diverse and very, very functional, which sort of illustrates the power of genetic modification, whether it be by simple gene transfers here, or potentially by gene editing to cure or at least have a very robust therapeutic effect in hematopoietic and immunological diseases. So another example of these severe combined immune deficiencies is adenosine deaminase deficient SCID, which is a metabolic disease. And around about the same time, same technologies were applied, very simple gamma retroviral vectors, and trials were conducted in several groups worldwide. And what you'll see here is that if you look at the number of patients treated, if you sum the different trial sites, there's 42 patients treated. The overall survival is 100%, which is remarkable, really, given that this is a very difficult disease to transplant at the best of times. And the disease-free survival using this rather imperfect technology was 74%. Now, the other thing I should point out here is that the vector used in the Milan trial was actually the first licensed gene therapy, ex vivo licensed gene therapy. So that's Strimvelis. So that was actually over the next 2 decades developed as Strimvelis. Now, of course, it wasn't all simple. You'll know that there were some toxic side effects of the gene therapy, and a number of these patients developed a leukemia as a result of the process. It took us a while to work out what it was, but it turned out that the vector configuration, which is a gamma retroviral vector with very potent enhancer sequences in it, was activating proto-oncogenes. And this is an example of one of the proto-oncogenes that was activated, in fact, in the majority of patients, which is LMO2. So this leukemic clone, this was from the one patient in the 10 patients treated in London. This leukemic clone had one single viral insertion site, which shows you the potency of mutagenesis, I guess, 30 kilobases upstream of this proto-oncogene LMO2. Now, there are plenty other mutations in this clone, but unrelated to gene transfer. So there's something to do with probably an acquisition of cooperating mutations through cell proliferation or during thymopoiesis. Anyway, we worked out that, we and others, I should say worldwide, worked out that the toxicity was because of the technology rather than any disease-specific issues, and that really meant that we could go back and try and fix it. And in doing that, what we had to do was to try and work out, which we hadn't done before, work out whether we could test whether the vectors that we were going to put into people were mutagenic. Could we say that actually this vector is safe? Could we say to a regulator this is safe? And that's one of the issues that we're going to come up against with the gene editing strategies. So the FDA and the MHRA in the UK will be asking the same question. How do you know that these, even if you see an off-target event, is that functionally important? And we spent a long time putting stuff into mice, and it really wasn't useful. And eventually we worked out there were some rather simple in vitro assays using primary cells, not using cell lines, that we could use to just test in a surrogate sort of way whether the vectors or the regulatory sequences in the vectors would cause a proliferative advantage to cells. So that's, we call it an in vitro mutagenesis assay. And that's pretty standard for hematopoietic gene therapies these days. It's not perfect by any means, but it is probably the only useful assay that we have at the moment. So on the basis of that, we were able to redesign the vectors, change the regulatory sequences, and conduct other clinical trials. And this is the second clinical trial we conducted for X-linked SCID, which is published, and we showed that, in fact, it had the same response as the first trial. The children treated responded very well. They developed normal numbers of T cells. And more importantly than that, to date, we've never seen any evidence of clinically manifesting insertional mutagenesis. And more than that, if you look molecularly for any signs of clonal disturbance in the peripheral blood, we don't see that. Okay. So again, technology moves on, and probably the most favored platform that you've heard about today a little bit for treating hematopoietic diseases now is the lentiviral platform, for reasons that they're probably safer because they have a slightly different insertion profile to the gamma retroviruses, And also, they're just better at getting genes into hematopoietic stem cells. So we spent a few years developing a lentiviral vector to treat adenosine deaminase deficient SCID. And I'm not going to go through the data here, but this is a summary of it as of, well, fairly recently. So we treated 48 patients with this vector now. Again, we see 100% survival in this cohort of patients. 47 of 48 patients with more than 6 months follow-up are able to discontinue their enzyme replacement therapy. I should have said that you can treat these patients with enzyme replacements. It's super expensive and actually pretty imperfect in terms of long-term immunological reconstitution. But suffice to say that most of these children have done very, very well, and most of them are able to come off all of their prophylactic treatments. So I think today, with this type of therapy, for this disease at least, we've now got choices, which is nice to have, particularly for the patients. So we have the choice of an allogeneic transplant, which has its issues in terms of toxicities, graft versus host disease, potential fatality. We have the potential for treatment with enzyme replacement, which is super expensive and imperfect. But also now we have an ex vivo autologous gene therapy, which to date has resulted in 100% survival and also near 100% efficacy. So here I think is a very good example of where we've got to the point over a decade, that's when we first designed the vector, to now where we almost have a standard of care from a gene therapy treatment. Now, what about, I just want to mention other stem cell gene therapies, because actually the landscape has broadened. And this is a list of different diseases, immunological, hematological, you've heard about thalassemia and sickle already, but also metabolic diseases that are tractable targets for lentiviral bone marrow gene therapy. And this list illustrates the diseases that are either in clinical trial right now at multiple sites across the world, or in preclinical development. Now, one of the points about the hematopoietic system is that it disseminates through all tissues. And this slide here is of a mouse brain where the little green dots are microglial cells that are derivatives of transduced hematopoietic stem cells. And the point I want to make with this slide is that, of course, those cells now can secrete enzymes or can secrete proteins that can treat multiple diseases. Now, actually, there's one disease where bone marrow transplant is ineffective, and that's metachromatic leukodystrophy. You just can't get enough of the enzyme to cross-correct this disease. With a gene addition strategy, of course, you can load up the cells with multiple copies of the transgene, and by doing that, actually load up the brain with corrective protein, which in this particular disease is aryl sulfatase A. So this data is from Alessandra Biffi from when she was in Milan. And what it shows you in this group of children who actually have a control group at the bottom of the left-hand graph, which is a sibling previously affected. And what you'll see that the gene therapy process now has allowed normal motor development and completely normal cognitive development. So I'll finish last couple of slides. Gene editing. Matt has showed very nicely that we can now edit hematopoietic stem and progenitor cells pretty well. There are issues about cell cycle because stem cells cycle less than progenitor cells. We can probably get around that, and we can probably make this into a scalable process. So that's why you are seeing a number of editing protocols in clinic now. What else can we use it for? Well, we can use it for diseases where actually the gene addition approach may be not quite so good as you want it to be because the regulatory sequences don't do what you want them to in the vector. And Wiskott-Aldrich syndrome is one where, although the immunological phenotype is completely corrected, the platelet phenotype may not. And actually, just to summarize this slide, what we've shown at least in vitro, is that if we can edit the Wiskott-Aldrich syndrome locus, then we can pretty much restore normal levels and normal physiological gene expression to these cells. Same is true of this other form of severe combined immunodeficiency due to interleukin-7 receptor alpha defects. Now, what about other cells? Well, T cells are obviously editable. In fact, they're more editable than hematopoietic stem cells. And of course, we've treated some patients with allogeneic T cells, which are edited to edit out the T cell receptor and CD52, to make them invisible to one of the immunosuppressive agents we use as a treatment for refractory B-cell leukemia. That's in combination with a CAR T. So T cells also very promising target for editing. So this is my last slide. I just want to say that the gene therapy process has evolved a lot over the last 2 decades. Editing is going to become part of the ecosystem very, very soon. The delivery of the edited cells to patients will, I suspect, follow very much the same lines as we've done for the gene addition strategies. The conditioning protocols will be similar, at least in the first instance. And I suspect the clinical responses will be equally as good. So I need to thank my many collaborators and also the funders of the work. Thank you very much.
Well, thank you, Adrian, for a very stimulating talk. You've heard now about a number of diseases from the many thousands that exist, single gene diseases. And we're going to hear next from Rodney Howell, who's been an advocate and a pioneer in screening for early effective genetic disorders in children. And he's going to give us some information about the current state of that art. So he's from the University of Miami. Thank you.
Thank you very much, Richard. I'm going to spend a little bit of time this afternoon discussing really 2 areas. One area of long-term interest of mine is Duchenne muscular dystrophy. That has been discussed widely as a potential candidate for genome editing, as this audience certainly knows. I'm not going to talk about editing. You'll hear a lot about that later. But I want to talk about Duchenne muscular dystrophy, the current state of affairs, what we know about it, what we need to know about it, and what we need to do about it. I'm going to end up then talking a little bit about newborn screening, which is going to be very critical for reasons that I will discuss later in the application of any of the novel therapies that we know about, plus the whole issue of genome editing. Duchenne muscular dystrophy was described by a French physician who was born more than 200 years ago. Interesting enough, he had a compelling interest in electrically stimulating muscles of the body, and he ended up mapping the musculature of the body extremely extensively and describing a lot of neuromuscular disease at that time, one of which he described as pseudohypertrophic muscular dystrophy, which today we call Duchenne muscular dystrophy after him. Duchenne muscular dystrophy is one of the more common fatal diseases, genetic diseases in childhood. It's X-linked and has an incidence between 1 in 4,000 and 1 in 5,000 boys. Which means that we will have about 500 new patients per year in the United States, and the incidence worldwide is really quite similar. The defect in Duchenne muscular dystrophy was discovered by Lou Kunkel in the early 1980s, and he determined that it was a deficiency of the protein that he named dystrophin, which happens to be the largest single protein in the human body. In spite of this groundbreaking discovery 30 years ago, it was a very long time before we had any specific treatments for Duchenne muscular dystrophy. I want to talk briefly also about the fact that Duchenne muscular dystrophy is also one of the best known diseases in the United States, and it's known because of long-term, big-time advocacy by the Muscular Dystrophy Association in the United States, which has advocated for treatment and so forth of Duchenne for a very long time. And in France, a comparable agency, a wonderful group, the AFM, based in Paris, that is extremely good and again has a great deal of information about Duchenne. Basically, that's very important and so forth. Now, if we look at treatment, I mean, in deciding whether or not something is really appropriate for editing, you need to say, is there an effective treatment? Corticosteroids have been used for a very long time. They're nonspecific, but they are clearly beneficial. They help both ambulation and long-term health and care. The work in recent years has really been looking and working on the mutations in Duchenne. And in a fairly recent 2015 TreatMD global database, there were more than 7,000 mutations that are known. And I've listed those here. I won't go through them, but suffice it to say that almost 5,000 of those were large deletions. And in addition to that, there are point mutations and other types of mutations, but there are a lot of deletions in Duchenne. There are 2 drugs that are currently marketed in the world that are approved either in Europe or in the United States specifically for Duchenne, and that is, one is that suppresses point mutations, and that's a small molecule that binds to the ribosome so that you get read-through of stop codons. That is called atalurin. That product, interestingly enough, is approved in Europe and has been before the FDA in the United States several times and is not approved in the United States. The second drug that's available is approved in the United States, and it's a clever drug that is an exon skipping drug. The one that's currently approved, it skips exon 51. And so it ends up getting the reading frame back in order and so forth. And that particular deletion is available in 13% of persons with muscular dystrophy. So about 13% of people in the United States have a specific drug. I want to make one comment as we're going along here, because one of the things that came up today, an important thing, had to do with the cost of the treatment for genome editing. And so we've been there and done that, because I can assure you that the exon skipping drug is as expensive as genome editing will be. It's extremely expensive. And so we've figured out how to get that done in the United States, which is currently working pretty well. So I've mentioned the fact that we have only one drug improved in the U.S. for exon 53, and its beneficial effects are not dramatic because it only does 13% of the people. However, in view of the fact that there are many, many other exons that are recognized, the company is obviously devising other exon skipping drugs, and so that group will go ahead. The gene therapy trials are going on widely in the United States. There are a number under trial at the current time, and they're quite impressive. And I think that we will see some of the gene therapy trials come to fruition and appeal to approval by the FDA within the next year. So they're really very good. So the bottom line is that the gene therapy trials, although they're working very well, they have a specific problem in that dystrophin is an enormous gene, and you can't get full-length dystrophin into the viral vector. And so the gene therapy trials are using many genes, which basically end up being very, very beneficial, but if successful, they will produce what we would call a Becker phenotype, which is a modest, it's a form of muscular dystrophy similar to Duchenne, except that you do much better and you survive much longer. So the point is, even with our most effective gene therapy, we still don't have trials that are really the kind that you would like to know about. Now, one of the things that is very widely discussed right now and currently in clinical trial is newborn screening for Duchenne muscular dystrophy. It was mentioned during the sickle cell discussion today that sickle cell disease is screened for in the United States. At the current time, Duchenne muscular dystrophy is not. Now, you might say, well, why would you want to screen for Duchenne? You diagnose it clinically. At the current time, the average age of diagnosis of boys with Duchenne muscular dystrophy in the United States is between 3 and 4 years of age. And the problem with that is that by the time you're symptomatic with Duchenne muscular dystrophy, you've lost an enormous amount of muscle. Because you've been losing muscle all this time, but you have sufficient reserve that it gets signed off and so forth. So the point is, it's very, very important that we have a technology that will diagnose these patients early, because all of the treatments we're talking about won't generate new muscle. They'll preserve the muscle we have, but if you've lost a lot of muscle, you've already lost a lot. So let me talk a little bit very briefly about newborn screening. And I'll talk about the United States, and if anybody has a question, I happen to be involved internationally in newborn screening. If you have questions, we'll talk about that later, but I'll focus on the United States. Newborn screening in the United States has been going on since the early 1970s. It even preceded the Asilomar conference that we've heard so much about before. The first condition screened for was phenylketonuria, which, if left untreated, produces profound developmental delay, and if treated carefully with diet, the persons turn out essentially normal. There are some problems they have, and I won't go into that, but the point is the treatment is dramatic, and since it was so dramatic, it was adopted throughout the United States, and so that the newborn screening program in the United States was very interesting in that it became a public health program, which is very unusual, because the newborn screening program in the United States is all under the aegis of state health departments. And every single baby in the United States gets screened for the conditions that are recommended, regardless of their ability to pay, because it has nothing to do with insurability. So that's one interesting thing, and one very, very exciting thing, and so forth. And over the years, other conditions were added to the panel, such as hypothyroidism and so forth. And since as this has evolved, the United States Congress, if you can imagine it, Congress has historically done something, but the U.S. Congress passed a law that requires the Secretary of HHS to have a national advisory committee that decides what should be on the newborn screening panel. And the other thing that's in the law, as written and signed by the President, is that the evidence that the committee provides, the recommendations the committee provides, must be evidence-based. They cannot be opinions. They have to be based on the best evidence, and so forth. I might point out we screen 4.1 million babies a year, making newborn screening very common. We screen babies, the big mechanism of screening are dried blood spots. And I show these on a piece of filter paper. This is an old slide, but the point is basically blood from the baby is spotted on these things. And then the filter paper is punched out. And today, the most common technology that's used is tandem mass spectroscopy that screens for a large number of things, but then other things can be done. And I might point out, we don't have any problems doing whole genome sequencing on these dried blood spots, which are also available, and we do a lot of genetic studies on them. At the current time, every baby in the United States, it's recommended by the federal panel, is screened for 35 conditions at birth. Now, most people think that their baby gets a PKU screen, but they get 35. And just for your own interest, I've just listed here what they are: organic acids, fatty acids, amino acids, hemoglobinopathies. We heard about sickle cell. But then there are lists of other disorders that are at the bottom: biotinidase, CF, galactosemia, GSD, severe combined immune deficiency, which has been screened for in the United States for quite a while, and our colleagues in the UK are coming up with that very soon. MPS, and the most recent addition is spinal muscular atrophy, et cetera. Now, Duchenne muscular dystrophy is fairly readily screened for, because babies who have muscular dystrophy, particularly Duchenne, on the dried blood spot, will have a dramatic elevation of the creatine kinase, which is the muscle-derived enzyme. And the screening test that's used now will be screening for the muscle isoenzyme of creatine kinase that improves its reliability. And babies with CK elevations, they are not modest, they're very dramatic. So the false positive rate is really very, very good and so forth. And so since the current treatments that we have available are mutation specific, when a baby has a positive CK, we recommend that they have whole genome sequencing. And the reason to do that is that if they have a mutation that's currently not treatable with the current drugs, it's likely that that treatment will come, and you don't want to go back and sequence or test the baby routinely and so forth. Now, it's likely we will soon have newborn screening, which will identify every baby in the country with Duchenne muscular dystrophy. Now, interestingly enough, the families, because of the reasons I've talked about, know a tremendous lot about Duchenne. Number one, even the less well-educated people do. They read about it, they hear about it. And the other thing is that the main fundraising organization for the U.S. MDA happens to be the firefighters. And the firefighters collect money every year in booths in the United States, and people trust firefighters. And so when the firefighters are out there talking about something, people take it quite seriously. If we say something, that's one thing, but the firefighters are very believable. But anyway, our families know about Duchenne, which is interesting. The thing is that although we have some beneficial treatments for muscular dystrophy Duchenne, we don't have any specific or curative treatment. And I must remind you that Duchenne muscular dystrophy is uniformly fatal. In the first slide, I didn't discuss it, but in 1960, the average age of survival was 14. It's now into the 20s. So you survive for a longer time, but you end up in a wheelchair and you die early, regardless of what good treatments you get. Now, the other thing is that our families also read a lot, and they read about genome editing. And this is the last comment I have, but I just got an email from a mother from India who is temporarily in Florida, and she wrote this email, and she says, "My son is suffering from Duchenne muscular dystrophy. I came to know this at age 4 because of the reasons I told you. That's when they're ordinarily diagnosed. He's currently 8 years old and able to do almost everything. I've read about the new technique called CRISPR-Cas9 to cure human diseases. Is there any way using the above technique for Duchenne muscular dystrophy? We are ready to do anything for him. Currently, he is in a very good condition, so please give him one chance." So thank you very much.
So our next talk is going to be by Charlie Gersbach from Duke University, who's going to tell us some more about genome editing as applied to some of these diseases. Charlie.
Okay, so thank you very much to the organizers for the opportunity to come and present some of our work that we've been doing. I was asked to talk about what we've accomplished in the area of applying gene editing to Duchenne muscular dystrophy, but I think that more broadly, I believe this might be the only talk or one of the only talks that will talk about in vivo somatic cell gene editing. And so what I'm going to try and do is use our work on DMD as an example for some of the broader issues that will show up in a variety of different in vivo somatic cell gene editing strategies. These are disclosures that I need to show, and the one that's going to be most relevant for this presentation is that a lot of our work in this CRISPR for Duchenne program is supported through a partnership with Sarepta Therapeutics. So we just got a fantastic overview of what Duchenne is and the effect that it has on patients. I think getting a little bit more into the molecular biology of this disease, it's important to understand that the mutation that causes the disease is to the gene that encodes this dystrophin protein that's shown up in the schematic. Dystrophin is a long linker protein that links the inside of the cell to the outside of the cell in different tissues, including skeletal and cardiac muscle. And that long linker has many different repeat domains or many different links to this chain. And they're shown here in yellow in the middle. And something that's important, and I'll come back to in a second, is that there's other forms of muscular dystrophy where rather than simply breaking that chain, it's actually a chain with a reduced number of links. So some of those repeats are missing. And because that linker is still present, but just shorter, it's still largely functional. And so a lot of the work in Duchenne is focused on restoring a shorter version of the protein that just includes a reduced number of those different repeat domains or different links in this chain. So why focus on Duchenne for gene editing? And we heard a little bit about that a minute ago, but I think there's a number of other reasons why Duchenne is a particularly, and neuromuscular disease in general, can be an interesting target for gene editing. We just heard about the dire clinical need and the limited therapeutic options for these patients, so I won't review that. But another interesting fact is that about 30% of all Duchenne cases are actually resulting from spontaneous mutations. So this means it's a spontaneous mutation in the egg cell that gets fertilized, and a spontaneous mutation in the X chromosome in that egg cell. And so these cases, and this is a large number of cases since Duchenne is one of the most, I think the most most common inherited genetic disease, are not applicable to germline editing, even if germline editing was safe and generally accepted. These cases still wouldn't be identified as for germline editing, since there actually is no carrier mutation in the somatic cells of the patients. So there's other technical reasons why Duchenne can be an interesting target for gene editing, including the fact that there are options for cell and gene delivery to the affected tissues, even though we are talking about correcting the genetic mutations through all of the muscle and heart tissue throughout the body. As we just heard, the full-length gene is too large to deliver with conventional gene therapy vectors, and so for that reason, it's advantageous to take advantage of the existing copy that already exists in the chromosomal DNA. There's a resident progenitor cells pool in skeletal muscle called the satellite cells, and we and others have demonstrated the ability to edit those satellite cells in vivo, and they might serve as a source to continuously regenerate the tissue with the corrected gene. And finally, because skeletal muscle is a multinucleated cell source where each cell has hundreds of different nuclei, we hypothesized at the beginning that potentially by correcting only a few of the nuclei, if editing wasn't particularly efficient, that even a small number of edited nuclei would be able to produce enough dystrophin protein to correct the whole muscle cell. And we have some data that supports that. And then finally, as we heard yesterday, homologous recombination and inserting or exchanging DNA in the genome is much less efficient than either breaking or removing DNA from the genome. And because of the nature of the genetic defect in Duchenne, there's actually the ability to correct the expression of a partially functional gene from its endogenous locus by removing DNA without having to exchange it. And to show you exactly what I mean, we focus primarily on this hotspot region that is mutated in the gene. And this schematic shows that the most common types of mutations that occur in Duchenne are actually deletions of one or more exons that make up, that are the chunks that make up the larger gene sequence. And so by focusing in on this region where many of the mutations are, we can actually use single gene editing reagents to address large numbers of patients. So this is an example of what that looks like. And each of these yellow segments is an exon, which is a segment of the gene that have to fit together in the right order. And so the different triangles and arrows represent how those segments of the gene fit together properly. And so one example of a common type of Duchenne mutation might be removal or loss of exons 48 and 49, or a deletion of 48 through 50. And so if you remove 48 through 50, as shown here, then you actually disrupt the reading frame of the gene, and the rest of the gene is out of frame, and you express no functional protein. However, if you remove 51 from this gene sequence, you can see that now those arrows fit together again properly. And so you get the expression of an internally truncated protein, but it's just missing some of those yellow linkers and can actually still function in linking the inside of the cell and the outside of the cell together. And actually, simply removing exon 51 can capture about 13% of all of the Duchenne patients. So to apply gene editing to this approach, we acquired muscle cells, skeletal muscle cells from a patient, a Duchenne patient, that was missing exons 48 through 50, and then we designed guide RNAs that would cut around exon 51. And if cutting simultaneously, you'd expect exon 51 to be removed, and in fact, that's what we showed in the cell culture experiment. We can sequence the DNA and show that it's the exact deletion that we intend to create, and then we can differentiate those cells in culture and show that when they upregulate the dystrophin protein, now the mRNA is missing exons 48 through 51. The parent population was missing exons 48 through 50, but now some of the RNA is missing 48 through 51. We can sequence it and show that it's the perfect junction that you would expect between 47 and 52, and by Western blot, now there's dystrophin protein being made in these patient cells. So I mentioned the removal of 51 can get to 13% of patients, but one of the nice attributes of this type of approach is that with 2 cuts in the DNA, you can actually cut out a much larger region of the gene, and you then lose more of the protein, but capture a much larger segment of the patient population. So for example, by removing all of exons 45 through 55, you get to about half, you can address about half of all of the different patient mutations. And in fact, that's what we're showing here, again, using the same cells that started out as missing exons 48 through 50, but now when we remove all of 45 through 55 with a single gene editing tool that could apply to half of the patient population, we can also restore some of the dystrophin protein. So all of that work was in cell culture. We then need to move forward actually applying this in vivo. So we moved to this mouse model where now we're going to remove exon 23 from this mouse, but it's the same idea where we'll be restoring the expression of a partially functional dystrophin protein. And so we made new CRISPR reagents that we can package into AAV vectors. Adeno-associated virus, or AAV, is our delivery vector of choice in this application. There are some pros and cons of using AAV, but really if you want to be able to get efficient delivery to all of the affected cells, skeletal and cardiac muscle throughout the body, this is really the only option that we have available to us right now. So we would inject that AAV virus either locally or systemically into this mouse that has this mutation to the dystrophin gene, and then we'd go back and look 8 weeks later for restoration of dystrophin. So if you take a cross-section of normal muscle tissue and stain those muscle fibers for the dystrophin protein, you see there's dystrophin coating every single one of these muscle fibers in a normal wild-type mouse. In the MDX mouse, which is the model of muscular dystrophy that we used for these studies, you see there's no dystrophin protein present. However, when we do a local injection of this AAV CRISPR into that muscle and then stain for dystrophin 8 weeks later, we see widespread dystrophin, and this is actually throughout that whole muscle tissue that we injected. We can quantify this. We see we get about 70% of all of the muscle fibers are now expressing some level of dystrophin protein, and that we suspected would be sufficient to restore muscle function in these tissues. And in fact, with Dongxing Duan at University of Missouri, whether it was twitch force, tetanic force, or looking at fatigue over repeated rounds of contraction, in all cases, these muscles got stronger in these mouse models. So that study was published a couple of years ago alongside other very similar studies from Eric Olson's lab and Amy Wager's lab. And since then, there's been a number of other studies from Jacques Tremblay, Jeff Chamberlain, Renzi Han, and others, basically doing very similar types of approaches. And I think that what I want to emphasize here is that in all of these cases, there was a mouse model of muscular dystrophy, and they did viral delivery of a CRISPR system that could restore and correct that mouse both biochemically and improve muscle function. But I think another important thing to point out is that all of the rest of the details of these studies were different. They used different models of muscular dystrophy, different mutations that they corrected, different Cas9 proteins, different guide RNAs, different AAV capsids, different doses of AAV. They treated the mice at different times. They looked at outcome measures at different times, different outcome measures. But in all cases, the kind of general story was the same, that the mice improved. And I think what's important about that is that the approach is very robust across lots of different labs and lots of systems, but also probably that there's lots of room for improvement, given that all those different parameters and variables that I just mentioned are probably important and could be optimized. Another study that stands out is in the bottom right, where it's a recent study from Eric Olson's group, where they actually used the same approach in a dog model of muscular dystrophy. And even though it's very early work with a small number of animals and looking at early time points, they did show dystrophin correction in many of the dog muscles. And so that's very exciting, and I think points out the direction that the field is going. But then in the last 2 minutes, I wanted to point out that a lot of those early studies, there was a lot of enthusiasm about gene editing for Duchenne, but also a lot of things that weren't really addressed in those early studies, that I think that the enthusiasm for gene editing was kind of predicated on a handful of assumptions that weren't actually looked at in detail in those studies. And so we've been working hard to try and address some of these outlying issues, and I'm going to talk about some of those here, although I don't have time to show data for all of them. But issue number one was that all of those studies were correcting mouse models of this disease, and the mutations that occur in most of those mouse models are not really relevant to the human mutations, and the sequence of many of the mouse genes is not homologous to the sequence of the human gene. So one of the things that the field has had to do is go and create new mouse models that carry human gene sequences so that we can actually test human gene-targeted CRISPR or gene editing reagents in small animal models. And so we and others have been going down that approach, and so we generated a new mouse that carries the human gene. We did that first. We created new patient-specific mutations by germline editing in the mouse, and then we could come back later with an AAV CRISPR approach and show that we could correct it just like we did the MDX mouse and restore some dystrophin expression. The second issue was that almost all of those studies looked at very short time points, and the whole the enthusiasm about gene editing is about the idea that we could correct these gene sequences and there would be a permanent correction for the lifetime of the animal or the patient, but that hadn't really been looked at in detail. And so we and others have now gone back and taken these mice out to well over a year and basically seen sustained and in some cases increased levels of dystrophin restoration and never seen any adverse events in these mice from getting a systemic treatment throughout all of their muscle and other tissues with the delivery of these AAV vectors. So the correction is sustained, but then that also begs the question that we assume that there's still some level of turnover of cells in these muscle tissues. And so if the cells are turning over, what cells are we actually editing, and how is the level of dystrophin correction being maintained? And in particular, we wanted to look at these stem cells of skeletal muscle that we know are participating in this cell turnover. These stem cells sit on the outside of the muscle fibers, they asymmetrically divide, and one of the daughter cells will then fuse into the muscle fiber, adding new nuclei. And so Amy Wager's lab had shown a couple of years ago that if you actually use a transgenic mice where these satellite cells are labeled with GFP, that she could see that her lab observed gene editing of these cells in vivo following AAV delivery, and we've now reproduced those results in our own lab, which is very reassuring, and shown that we can isolate those satellite cells, and when we deliver the CRISPR system, again with a variety of different AAV vectors, that those GFP cells now have the editing event that we intended, in this case removal of exon 23 from the MDX mouse. Now, another issue that gets routinely brought up for these types of in vivo genome editing approaches is the potential for an immune response to the different things that are being delivered. And other studies have already addressed immune response to the AAV viral vectors that are commonly being used. Previous studies have looked at potential immune responses to the neoantigens from the restored dystrophin protein that otherwise were not present in the animals or eventually in patients. But one of the things that we were particularly concerned about was the immune response to the Cas9 protein that is a bacterially derived protein that we're now delivering systemically into these mice. So I want to, again, remind you that we saw long-term expression of dystrophin for over a year and never any adverse events in these mice. However, when we did draw serum from these mice, we were readily able to see antibodies against Cas9. That's not surprising, and others in the field had described similar results. Of course, because the Cas9 is always going to be intracellular, we're not actually that worried about the antibodies against Cas9 and would be more worried about a T cell response to peptides that would be displayed by cells expressing Cas9. And we do, in fact, see T cells by interferon gamma ELISpot assay only in adult mice that we treat. And so we still need to have more work to do to understand how we resolve the fact that we see basically no adverse responses and long-term dystrophin correction, but also potent immune responses. I'll also point out that in these studies, we were using a ubiquitously expressed constitutive promoter, and one way that we might be able to address this moving forward is to restrict the expression of certain tissue types. And then finally, a concern that we've had that's been discussed throughout the course of today is what's going on at the on-target site and what's going on with the stability of the different vectors that we're delivering. And we were particularly interested in this because of, as was discussed this morning, kind of an unexpected result that's been described over the last year of very large deletions being created at the locus, at the on-target site that aren't picked up by your typical assays for looking at gene editing at the on-target. And so we developed a strategy that would allow us to do an unbiased assay, just sequencing into the break site to see what was going on, looking for other things than the intended event. And we actually were able to reproduce this observation only in liver tissues in these mice, not in the skeletal or cardiac muscle, but we were able to observe these large multi-kilobase deletions at the target site. But we also looked for other types of events. So the other things that we might potentially observe would be inversions or indels at the on-target site without the intended deletion, translocations, or something else that we were concerned about was AAV vector integration into the break site. And so I don't have time to walk through that results and show here, but I'll just point out that we see a lot of other types of events happening at that on-target break than the intended deletion that we want to create. And one of the most frequent is actually integration of the AAV vector that is carrying the CRISPR-Cas9 system into the break point in the dystrophin gene. Now, importantly, we don't think that there's any reason to think that this would cause adverse consequences, at least so far. But it does take away from efficiency of getting the intended event that we're looking for, which is the removal of one or more exons. And I'll also point out that it had been reported previously, this isn't, David Russell's lab had shown that AAV vectors do integrate very efficiently into chromosomal break sites, so it shouldn't be unexpected that it would jump into these breaks that are being created by the CRISPR system. But it also occurred to us that if it's integrating very efficiently at the on-target site, it might also be integrating at other sites across the genome where breaks are getting made. So we then went back to the same samples and sequenced, rather than into the on-target site in the dystrophin gene, we sequenced out of the AAV vector. And we could see that there's a very efficient recovery of sites where AAV integrated in the on-target site, but we could also find integrations of AAV vector into other sites across the genome that we could then go back and map and find pseudo-guide RNA target sites underneath those peaks. And it's important to point out that some of those guide RNA target sites we had predicted bioinformatically, but could not detect any activity by conventional deep amplicon sequencing assays, but we were able to recover them by sequencing out of these AAV vector integrations. So this is the last slide to summarize. There's a variety of approaches being explored for genome editing for Duchenne. In all cases, they primarily focus on removing gene segments to restore a functional but partially truncated dystrophin gene rather than restoring a full-length normal protein. Many studies now are reporting long-term dystrophin expression with no reported adverse events. A couple of groups now have reported relatively efficient editing of the adult stem cells of skeletal muscle. But we also do see robust anti-Cas9 immune responses and unintended events at both the on-target and off-target site. And I think that these really need to be the focus of future studies. And I'll just leave this slide up to thank the collaborators and students and postdocs that were involved in the work and the funding sources. Thank you.
Well, thank you for a very informative talk. Now, as you've heard earlier today, and I think yesterday, it's important in thinking about all of these things to think about the relationships between the patients that one is hoping to treat and the scientists who are hoping to treat them. And our next speaker is Pauline McCormack from the University of Newcastle, who's a sociologist who's been very interested in interactions with patients, informed consent, and various other aspects of the relationship between the patient population and the scientists. So she's going to tell us about that. Pauline.
Thank you, Richard. I'm delighted to be here, and I'd like to thank the organisers for inviting me. I've got a medical enhancement in the form of new glasses, but if it has an unintended effect, then I'll swap to the old model. So. Yep. Thank you. As you can see, the title of my talk is Gene Therapy for Duchenne Muscular Dystrophy, or DMD, The Long and Winding Road. And it fits in quite nicely to what's just gone before. I'm not even sure I'm going to talk that much about genome editing today. Instead, I'm going to provide a tale about the swerves, the detours, the roadblocks that might appear on the translational road for genome editing. When I see talks about the ethics and the social effects, they're very much generalised. So here I'm just trying to be more specific by using development of therapies for a particular disorder. We don't know what it's going to look like on the translational phase, so I want to ask if this is an example that we might generalise from to other disorders, to translating genome editing for DMD. So Rodney covered the aspects of the disorder, but I do want to stress that it's severe, progressive and life-limiting. And if I just concentrate on the high care burden on the family, I mean, there are some studies that show good quality of life and an increase in family cohesion for Duchenne, but typically families report care burden and support issues around isolation, social inequality, psychological stress and depression and destructive behaviour. And caregiver burden gets worse as the disease progresses. Siblings of children with DMD say they receive less parental time and attention and they miss out on activities. So the story I want to tell is, starts going on for 10 years ago. I was in a room with international researchers, clinicians, patient advocates and regulators at the European Medicines Agency in London to talk about gene therapy for Duchenne. Trials in humans for a technology called exon skipping, which you've heard about, were just starting, and we were there to talk about possible regulatory barriers. So the dystrophin gene you've heard, which codes for protein which builds muscle, is the biggest in the body. And Duchenne is caused by the mutations in this gene. It's been suggested up to 7,000 I heard today. I'd previously heard 3,000 different combinations. There are different types of mutation and they appear at different points on the gene. So exon skipping therapies that we were there to discuss were targeted at one particular point of mutation. Once the first therapy was successful and licensed, subsequent therapies would be partly the same and partly different. So the meeting was convened with the EMA and representation from the FDA to discuss how further therapies could be licensed. When the first one was successful, how did we get the rest out quickly to patients? I think we talked about being cautious, but there was a really high expectation that these therapies would be successful. They showed such promising results in animal models. And I don't think any of us in that room could have anticipated just how slow and involved the journey towards providing the therapies would be. And I'm suggesting that for some disorders, possibly for Duchenne, the genome editing translational road could be just as slow and just as involved. Really, we should have had an idea. Viner et al. In a recent article looked at the discourse and developments around the geneticization of society debate over the past 25 years or so. And the authors concluded that the specific expectations around geneticization have created a powerful genetic imaginary. Referencing Jasanoff, they say, an imaginary is a collectively embraced, actionable future in which technological change will bring about a positive, culturally intelligible result. And that's really what we were thinking in that room 10 years ago, and partly what a lot of people have been talking about over the past couple of days. And assumptions are often made that novel technologies will map directly onto the real-world experience in a predictable and linear fashion. But history tells us that the application of new health technologies is very rarely one of stepwise improvements and a reduction in uncertainty. Discrepancies between the promised benefits of the genetic imaginary and actual experience can then lead to unintended consequences, lack of trust, disappointment, and then lack of consensus for everyone involved. And that then can be disruptive to the actual technology itself. For sociologists like me, when we talk about genome editing, it's abstract. We have no idea how it might play out in the real world, and really no idea of how it might benefit or not benefit people with Duchenne and other families with rare diseases. Margaret Sleebon-Faulkner referred to it yesterday as a risk of overestimation of disembodied knowledge. So this has definitely been the case in terms of gene therapy for Duchenne over the past 10 years. There weren't stepwise improvements, there wasn't a reduction in uncertainty. Instead, there were multiple problems and a massive increase in complexity. Some of them were things that the scientists might have surmised about, but others were completely new. And some of them we may be able to work around, but some of them are obstacles that are just not going to be moved. We've already heard that we're dealing with the largest gene and we're dealing with thousands of mutations, so you're starting out with that level of complexity. But then there was an immune response to the delivery vehicle. There was an unpredictable phenotypic effect. In one trial, I remember there were these real outliers, kind of super responders, but the trial itself didn't meet its endpoints. The distress and anger of the parents of those responders was a really difficult thing to watch when they found out that the trial was unsuccessful. And then there were all sorts of complexities around suitable primary endpoints, difficulties establishing biomarkers, and what constitutes benefit. Just taking something like the primary endpoint, which was a 6-minute walk test, that itself took years to develop and to validate. And then in practice, it just didn't deliver as we expected it to. So I'll paraphrase Francesco Montoni, who's one of the leading clinicians on this, and he says, "We were once optimistic, but the results are pessimistic." So where are we now? As you've heard, there are 2 therapies available. One is an exon-skipping drug. One's available under accelerated approval in the US, but it's been refused by the EMA. The other's licensed in the EU, but it's in the UK, for example, it's on managed access until they gather more data, and it's not available in the USA. And this is because regulators have concluded that clinical benefit is not proven. Hoffman and McNally, who are clinicians who work on this, sum it up thus: the current situation serves to illustrate the shifting sands and blind alleys that may disorientate the modern orphan drug developer, as well as the families anxious for a drug that can slow the tragic progression of Duchenne. So we all know that 85% of late RCTs in humans fail. But we're not talking about genome editing in those terms, or with a view cast back to the long winding road that many therapies take. Instead, we're talking about genome editing in really positive tones. We talk about its enormous positive potential. And for DMD, it's been said that it has great hope. And it might be that some of the complexities can be overcome. Charlie's just outlined how they're trying to anticipate a lot of those complexities. Other scholars say that what was the disadvantage, the size of the dystrophin gene, in gene therapy may be an advantage in genome editing. So directly quoting Ellen Ripley from Aliens, I would like to say, I hope you're right. I really do. I think there's a typo with the date there, but the Nuffield Council on Bioethics, in their report on human genome editing, said that it's okay to be aspirational or to talk about promissory futures. What is dangerous is where there are asymmetries of power, information or representation in the public sphere that mean that certain visions and values go unappreciated and others go unchallenged. To me, one aspect that goes unappreciated is this. The single thing that's improved quality of life for people with Duchenne and has extended life expectancy, it's going on for doubling it in 25, 30 years, is care. And we don't talk about care. This is what's made the most difference. And on asymmetries of power, this was touched on this morning and yesterday by Dana Carroll. One of the problems for rare disease is access to therapies. There just is not equity of access, and this is not a North global South problem. So Eurodis, the umbrella European Organisation for Rare Diseases, in 2011 did a survey of the EU member states, and these are not poor countries. And they found that in over 21 of the 30 EU member states were only providing compassionate use access to orphan products in about 10 to 20% of cases. So tiny proportions of the therapies that are actually available are getting to patients. And with the existence of real-life problems like this, you can see why I view gene editing reaching individuals as conceptual. And although I've used gene therapy for Duchenne as an example, if we zoom out a bit, I've had a little bit of involvement with the 100,000 Genomes project in the UK. And this is moving directly towards clinical application of whole genome sequencing in the NHS. And we hear from people involved in that similar uncertainty and complexity is introduced at the same time that the government and other authorities are saying we must push towards this genomic dream. So again, the message and the experience are divergent. So, in terms of genome editing for Duchenne, I would say let's press on, but with some more cautious language. People with rare disease and their families deserve what you might call a decent genetic minimum. I'd love to see the care burden removed or ameliorated for Duchenne or other rare diseases. And despite all the setbacks in gene therapy trials, they've given us an awful lot of information and an absolutely excellent trial infrastructure. But we have to remember that success in the lab is not success in real life for people with rare disease. And I think we need to strike a more nuanced balance between aspiration and making promises that we can't keep. We risk over-promising and under-delivering. And that's what we did in gene therapy, with no idea about the length or the arduousness of the journey towards success. I think we should under-promise and then we might over-deliver. Wouldn't that be great? I think we do ourselves and the people living with genetic disease a favour if we anticipate a long and winding road. Thank you.
Well, thank you. So we're ready to take questions and have a discussion. So if you have questions, get them ready. Let me just start off with Pauline, actually. So you make a good point about the expectation versus delivery issue. So do you have any concrete suggestions about how to improve this interface so that there is more understanding on both sides of the issues facing the other? Is there some concrete suggestion that one could implement?
Well, I do appreciate that it is a difficult thing to do, and that in order to gain acceptance of technologies and funding and awareness, that you do need to talk about the fact that it could be positive or it has positive potential. I think relationships between patients and clinicians, we're getting much better at that, but we still deal with a group of very educated, quite active patient advocates. And I think we can pay more attention to how we put our messages out, how we can tell them in a more straightforward way that will allow us to bring more nuance. And there is an engagement session tomorrow, so I think everybody here should attend that and learn from it.
Alta.
Alta Schauer from the University of Wisconsin. First, thanks very much. It was a wonderful session. With due respect to the due care, I want to focus on the science for the moment in the question. Dr. Howell, you pointed out the value of early diagnosis in order to begin some kind of therapy for muscle sparing. It made me wonder whether this could be done even earlier. Is it possible to do this kind of diagnosis in utero? Dr. Gersbach and both of you were talking about the large number of mutations that account for this particular disorder. So I wasn't sure if you could actually identify it that early, and if so, would gene editing in utero ever be a feasible approach to this?
Let me make several comments. Number one, Charlie has pointed out a couple things, is that most babies are born with Duchenne without a family history and without the family knowing about it. So it's a surprise. And the second thing is a third of the patients are new mutations, so that there's no, so there'd
be no reason for the
So
screening.
you have that situation. But you clearly can diagnose Duchenne in utero, and that's been done in families that have had previous children and so forth. And I would leave it up to Charlie about whether or not you think that I would think there's no reason you couldn't do gene editing in utero, but that's not my business.
Actually, I would ask you a question about the, just following up on the in utero diagnosis. I assume that CK would not work in utero, but a sequencing approach would work.
I would do sequencing and so forth, yeah.
You'd have to do sequencing.
Yeah.
So in utero gene editing, just like in utero gene therapy, has been explored in preclinical models. It's definitely been done in, it's been tested in mice, and I think there's some examples, certainly for gene therapy, maybe even for gene editing in large animal models. I think it's fraught with all the technical, ethical, moral complications, practical complications that germline editing and other things are. But technically, it's feasible, and just like there's some of the practical advantages of germline editing compared to somatic cell editing. Once you've got more cells and more tissue and a fully developed immune system, the same thing applies to the in utero approach, that less cells, less tissue, circumvent some immune system issues. So there are some advantages to the approach.
Hi, Santiago Molina. I'm a PhD candidate in sociology, and I had at UC Berkeley. My question is kind of about the panelists here, and to Professor McCormack specifically. I guess I'm kind of interested in what the role is of the social sciences in helping to produce or helping facilitate the interactions between patients and basic researchers. And the reason I ask that is because I'm a sociologist, but I'm also in a lab in Jacob Korn's lab. And so I guess I'm wondering what models you guys have used for putting together the panel, and what other opportunities are there for more involvement of the social sciences in research.
Well, we didn't put together the panel, so I'm not quite sure whether we can answer that. So I think maybe you're getting at the question which relates to what I asked Pauline. I think that Pauline's answer was that this is a well-informed patient group, and I believe that's true of many of these genetically inherited diseases. You alluded to it actually earlier. It seems to me there must be ways of taking advantage of that better than we currently do.
So I think one of the ways is that social scientists can help is in understanding the theory around societal interactions. But for me, one of the areas that's been most helpful for me was my experience in education. So when I came to work in large groups whereby it's already difficult for the people who are doing this full-time to talk to each other when you're doing something like a bioinformatics project, because you've got a geneticist here and a computing scientist here, and a database programmer there. So it's about creating the correct spaces. It's about making sure that the collaborations are set up so that first of all, everybody is willing to listen to everybody else. Secondly, no one person is the expert in the room, and that includes the patients. So you have to be willing to stand back and say, everybody here has something to teach me, something I can learn from. And the role of the social scientist can be in using their knowledge of theory to create those spaces, making sure those spaces are used, making sure the relationships are working and are healthy, and reporting back on that to everybody concerned. I hope that makes sense.
Yeah. Thank you.
Let me add one thing, is that I think it's really important, however, for families who have a rare disease, particularly a fatal rare disease, to maintain hope. And so hope is really important to the family. So while you're being realistic about what's going to happen, it's very important, I think, to be sure that you keep up the hope, because that's really what keeps people going.
Thank you.
So I have a technical question for Charlie. So when you're doing in vivo gene editing for muscular dystrophy, you want to target both the muscles and the heart. Is the choice of the virus you use to deliver, does that bias one way or another? And also, those are two very different sorts of tissue. As you said, muscle is a syncytium, and so that getting a few genes, a few nuclei edited might be good enough. Heart is not. You have to get into each cell, or maybe you can make do with only some of the cells being edited. What's known about that?
Yes. Yeah, so that kind of motivates our use of the AAV vectors, the adeno-associated virus, because before any of this work, it had been shown that AAV9 in particular can be delivered systemically by intravenous administration and get to virtually all of the skeletal and cardiac muscle. Now, it also goes to a lot of other places as well. It's also used to go to the CNS. Most of it goes to a large amount of it goes to the liver, and it probably goes to some other cell types as well when delivered systemically, but it also is very good at getting to all of the skeletal and cardiac muscle. So, and it's really the only delivery vector that I think has been described that can do that. So I think if you take that vector for delivery, but then layer on some other ways to restrict expression, like a promoter that is on in both muscle and heart, then you can hopefully get the outcome that you're looking for. But there's some other technical nuances. But
So this is a particularly good example of delivery in vivo, or are there more coming along the same lines for other tissues?
Are there disease indications, or? Well, I think some of the ongoing trials were summarized on the first day. What's moved forward first in vivo is editing in the liver, and there's Sangamo has a couple of clinical trials, and they're using AAV vectors in that case. There was announced an IND filing to do gene editing in the retina that presumably might start in the next year, and that's also using AAV vectors. So most of the stuff that's out there right now is using AAV vectors for delivery. The different serotype and capsid proteins varies depending on the tissue type. But the approach is generally similar. And then there are some other non-viral approaches using lipid nanoparticles that will be tried first for gene editing in the liver, and they're staged a little bit further behind what's going on with the viral vectors.
Thank you.
Okay, Komi Tani with Saitama Medical University in Japan. I also have in vivo genome editing stuff for Cherry. I have a question. So after in vivo delivery, how would you evaluate the off-targets which might happen in human? Even though you have very nice humanized mouse model, the mouse genomes are quite different from human. So if you have any
Yeah, so the question is, how do we look at off-target effects in vivo? And there's a couple of ways that we've been doing that. One is to do bioinformatic prediction and then go in and do targeted deep sequencing of all of those different sites. So we've done that. Another approach would be to do a kind of comprehensive analysis in a cell line where using something like GuideSeq and then looking, and that way you could use it you could do it in human cells. Obviously, that doesn't get to the in vivo question. Another approach that we've used is the CircleSeq approach, which is an approach where you look at CRISPR cutting of the genome outside of cells with purified genomic DNA, and we can do that with purified DNA, find any potential off-target sites, and then go and evaluate them one at a time in vivo, and we've done that as well. And all of these are kind of effective at getting to different aspects of the off-target question. But most recently, this approach of looking at AAV vector integrations has been very sensitive at looking for off-target effects that off-target DNA breaks that we were not able to recover by kind of any of these other methods. So I think that when using at least AAV vector delivery, I think sequencing out of your AAV vector and seeing where is it going in the genome could be really powerful for finding off targets.
How you know that what might happen in human patients after delivery? It gets hematopoietic cells. You can transplant all the human hematopoietic cells back into NSG mice and see what happens. But in muscle or other, tissue in vivo, it might be difficult to predict what might happen.
So there are some options there. So I think the question is that you can do in vivo delivery to a mouse or a primate, but if the sequences are different, then how do you know that it won't find an off-target site in the human genome, even if there's no off-target site in a primate or mouse genome? And one way to handle that is with explanted human tissues. And so, for example, there are mice that have been engrafted with human liver cells and basically repopulate the whole mouse liver with human hepatocytes. That's one approach for doing in vivo delivery to human tissues and looking for off-target effects. I believe that in some of the studies that are moving towards the clinic with gene editing in the retina, they can take cadaver retinal tissues and test for editing in those explanted whole eye tissues. So, and there's similar approaches that you could do with muscle, where you can engraft animals, immunodeficient animals, with human muscle cells. But I think that the efficiency isn't quite at the point you would need it to be able to robustly look at all off-target editing events.
Thank you.
Yes, Seigo Izumo, Takeda Pharmaceutical. I have follow-up question on AAV vector and the integration. I thought that AAV was generally touted as episomal and doesn't integrate, but is this something that people overlooked, or if you combine AAV and the Cas9, preferentially, there's a new phenomenon that because of a combination with a nuclease.
Right, so there's a body of literature describing this, but I think that it's not maybe widely discussed. So AAV vectors, the vast majority of the AAV vector does stay episomal, but a small amount of it does integrate into the genome, and the estimates are somewhere around 1% or less. But if you're talking about 1% of all of the millions or billions of cells that might get transduced with AAV vectors, it's still a large number. And the dominant way that, so the AAV vector is a linear double-stranded genome, and it will jump in to double-strand breaks, and it appears to do so through something that looks like an NHEJ-based mechanism. So even if there's no DNA breaks, there's no gene editing tool, and you just deliver an AAV vector like a conventional gene therapy, like many that are out there right now, some of that AAV vector does jump into the genome. And it does so largely at spontaneous double-strand breaks, because we get spontaneous breaks in our cells all the time, but the cell's very good at putting them back together. If there's an AAV genome sitting around, it might jump into that break. So even without CRISPR or any gene editing tools, you will be able to find those, and we see those as well. And I think that happens in the clinical protocols that are using AAV gene therapy. Now, if you intentionally and very efficiently create a break with like CRISPR or another gene editing tool, then I think that the vector will, at an even higher efficiency, jump into that dominant breaking site. And so I am of the impression, and I think there's other data out there that would support this, that any of the gene editing approaches that include a double-strand break and an AAV vector, there is some amount of that AAV vector jumping into that break.
Thank you. The other question is, you mentioned there's a robust antibody response, immunogenic response to, Cas9, or are you saying this is, many people have Cas9 antibody, I was told at the beginning. So you are putting in, so even though there's a strong antibody response, that nothing clinically happens?
Right, so in these mice, we believe there doesn't appear to be any pre-existing immunity in these mice. I think in some animal populations there is pre-existing immunity to these Cas9 systems, but our measurements suggest that that is not the case in these mice when we're treating them. But we do see this induced response. What I'm saying is that we do observe a response, but it doesn't seem to be enough to cause an adverse event. It doesn't seem to be enough to clear the edited cells, and it seems to resolve itself autonomously. So I think we still don't understand this relationship of the immune response and the cells. And we're not, I'll emphasize, because we're not delivering Cas9 protein directly, we're not really worried about the antibodies. It's the T cell responses that would be more concerning. And in some studies, not the ones that I presented, we've observed a transient elevation of liver enzymes that would suggest a T cell response to Cas9 expressed in the liver, and then they self-resolve, and the mice are fine, and the livers are fine, and edited cells remain. So I think it's something that just requires more analysis, but isn't necessarily a showstopper at this point.
Well, thank you. If there are no more questions, I'd just like to thank the speakers for excellent presentations and admirable discipline keeping on time. We've made up some of the time, so there's a break now, and I believe we should be able to get back on schedule starting the next session at 4:15.